Hi students, this is Ms. Shim, and today I'm going to be talking about the structure and function of large biological molecules that make up living things. Okay, to be honest, I'll just be honest right now, as I'm feeling a bit tired. I just came back from my son's soccer game. It's really hot today, and so I hope that this video goes okay and that I make some sense. I almost feel like I can just fall asleep during this video, and probably some of you will fall asleep during this video. I hope it's not too long. It is a big topic. It's a big topic, a lot of things to cover. So this video is probably going to be long, and just remember, you don't have to watch this at 1x speed. You can watch it at 2x speed, but hopefully you guys won't fall asleep. I feel like falling asleep. I'm super tired. All right, so in our textbook, it's going to be chapter 5. Let me make myself a little bit smaller. And again, the topic is the structure and function of macromolecules. So that word macromolecules means large molecules. And so living things are made up of large molecules. And there are 4 types of large molecules. We have the carbohydrates, the lipids, proteins, and nucleic acids. And we're going to be going into each category. Okay, so macromolecules, and macromolecules meaning large molecules. So these are large molecules made up of small parts. And how do these parts come together? By a reaction called dehydration synthesis. Okay, so when I say macromolecules, which are made up of smaller molecules, the small molecules are called monomers. So like a monomer, okay, a monomer means one piece, okay, whereas a polymer means many, okay, like many pieces. Think of it like, you know, like Legos, right? Legos, each little piece comes together to make, I don't know, like a Star Wars figure or whatever, okay? So with polymers or macromolecules, like carbohydrates, how does each piece come together? There's a reaction called dehydration synthesis. In some older textbooks, they're called condensation reactions, okay? But dehydration synthesis is the main way of describing how these polymers are made when the monomers join together. So let's go over that. Dehydration means to lose water, right? Like, so when you're dehydrated, you've lost water. Like, I feel really dehydrated today. Today's a really hot day and I was out in the sun. And so water is going to be lost, or water is going to come out. Okay, water is going to be lost. It's going to come out of the molecules. So in this example, I have a really short chain, a short polymer. This polymer is only made up of 3 pieces, or like Lego pieces. All right, and then if I want to add one more, so if this is going to be the 4th one, how does the 4th one link up with the 3rd and make a longer polymer? How does it link up and become a longer polymer? Well, this hydrogen atom, which is attached to the 3rd monomer, and this hydroxyl group, remember functional group, hydroxyl group, which is attached to this 4th monomer, together H2O, they make H2O, and H2O is going to come out. It's going to be lost by these molecules, and now molecule number 3 and 4, they're going to form a covalent bond. Here's our covalent bond. A covalent bond is made, and now we have a longer polymer because water has been lost. Water is removed in a dehydration synthesis reaction. Okay? All right. So that's dehydration synthesis. That's how we make polymers, macromolecules, like macromolecule meaning large. How do we go from like single pieces to something bigger? By dehydration synthesis reaction. And that's super important. You need to remember that for the whole year, okay? All right, so on this slide you can see here, this is an example of how a disaccharide is formed. Okay, a disaccharide. Sorry, my writing is so messy. Di is the prefix that means 2. Saccharide means sugar. We have a monomer here called a monosaccharide. All right, so this is a monosaccharide, and then here's another monosaccharide. So we have two monomers. Example is glucose and fructose. How are they going to come together to make a disaccharide? Well, dehydration synthesis. This hydroxyl group, this hydrogen from this hydroxyl group is going to come out. Together they're going to join and make water. Dehydration. Water is lost from the molecules. Water is removed, okay? And now this oxygen is going to make a covalent bond with that carbon, okay? So that carbon on that glucose molecule, they're going to form, that carbon right here is carbon number 1, and it's going to form a covalent bond with this oxygen, which is connected to this carbon on the fructose molecule, and this is carbon number 2 on the fructose molecule. So we call this a 1,2 glycosidic linkage. Glycosidic refers to sugars, so it's a covalent bond that links two monosaccharides, two single sugars together. So when two single sugars come together, they're called glycosidic bonds or glycosidic linkages. Okay, these are strong covalent bonds. And then when you add another, right, when you add another monosaccharide over here, they're going to form a glycosidic linkage. And again, it's by dehydration synthesis where water is going to be removed. Okay. So then when we eat sugars, okay, so when we eat long chains of polysaccharides, so let's say that you're eating toast for breakfast, cereal for breakfast, there's a lot of sugar in there. We're going to break it down by hydrolysis. Okay, hydrolysis is the opposite of dehydration synthesis. Hydrolysis. So what is, let's break down this word. Hydro refers to water, H2O. Lysis means to break. Okay, so in this example, it actually can refer to two things. Water itself is breaking in hydrolysis, but also we're breaking down the polymer using water. So there's like two meanings here. Hydrolysis is where water breaks, but then also we're using water to break the bonds in a polymer. So in this example, we have 4 monomers joined together when we add water to this covalent bond. Okay, H2O is going to break apart. You got the H on this side, and then you got the OH on this side, or HO, hydroxyl group, which is going to attach itself to monomer number 4. Okay, I was going to say something, but I forgot. It's okay. I'll probably remember later. So carbohydrates. So remember, there's 4 macromolecules that make up living things. Carbohydrates is the first one. Carbohydrates are sugars, okay, sugars, and long chains of sugars or polymers of sugars. The primary purpose of carbohydrates is to store energy, okay? It's an energy source or to store energy, and also it's going to be a building block, okay? And the formula for sugars is actually CnH2nO, N. So N being anywhere between 3 to 9. Okay, so carbohydrates are monosaccharides, I should say. Monosaccharides, monomers of sugars. This is the chemical formula. So if N was 3, then you would have a sugar that is C3H6O3. If N is 6, C6H12O6. Okay, using this formula. And then all sugars are going to end in OSE. So whenever you see OSE at the end of the molecule's name, the name of the molecule, OSE, you know it's a sugar. Like glucose, fructose, sucrose, lactose. You might see in your textbook sugars shown as a branching chain. I'm sorry, yeah, as a chain, I should say, a straight chain. Most of the time, though, in nature, you do not have sugars that are going to be in a chain. They're going to be in a ring form. But I just want to bring your attention to this. Remember, we talked about the equation for sugars. If N is 6, then the chemical equation is going to be C6H12O6. If N is 5, it's going to be C5H10O5. Okay? All right, so as I was saying, you're going to find sugars in a ring format in nature. Okay, they are not commonly found in this straight chain format. And it's just showing you how it folds into a ring. It's just more stable as a ring structure. And this is carbon number 1, right? This drawing right here is the abbreviated form where they're not showing you where the carbons are, but every point here is a carbon. Okay, this is carbon number 1, 2, 3, 4, 5, 6, going in a clockwise direction, starting with this oxygen. So this corner, there's no carbon. That's why there's oxygen written in there. But this is an example of glucose. It is the most common monosaccharide that's found on Earth. And so I was saying earlier, how do they form? How does a disaccharide form? By dehydration synthesis. And you can see that OH plus H, water is removed. Okay, and you get maltose. Maltose is a disaccharide made up of 2 glucose monosaccharides. And then over here, and kind of blocking it, but this is sucrose. Sucrose is table sugar. It's the sugar that you have at home. You mix with your tea, coffee, you bake with, and it's made from glucose and fructose. When glucose, fructose come together by dehydration synthesis, water is removed, they form a glycosidic bond. Okay? So then poly, right? Poly means many, saccharide means sugar. And so there's 2 main functions or purposes of polysaccharides. Let's talk about the one in plants. Okay? In plants, an example would be starch. Okay, in plants, the example is starch. It's a long chain of polysaccharides that's going to be used to store energy. So think about a potato. A potato stores energy. Okay, and I left it at school, but I do have a really old potato at school, and it's like growing, like from the little eyes of the potato, all these roots and stems and leaves are even growing out of it, because the potato itself is energy storage for growth for the plant. Okay. And so this is starch. Okay, you can see here each hexagon represents a glucose molecule or monosaccharide. They're all linked together. Amylose is a long chain, and then amylopectin that also makes up starch is a branching chain. Okay, so starch that's found in plants for energy storage, there's like 2 forms of starch. There's amylose and amylopectin. They make up starch. In animals, looks very similar. Okay, we also, in animals like us, we also have long chains of glucose molecules, but we call that glycogen. Okay, glycogen. And glycogen is energy storage for us as well. We store glycogen in our muscles and in our liver. Okay, and one of the reasons why we store glycogen in our muscles and liver is because those are some of the most active parts of our body, right? Like, so let's say that you have to run, right? If you have to run, if your life is in danger and you got to run and get away, there's immediate energy that's being stored in the muscles that can be utilized, right? So that's polysaccharides, the first function, which is to store energy. In plants, it was called starch. In animals, it's called glycogen. The second role or job of carbohydrates is structural purposes. It's to provide structure for the organism. In plants, it's called cellulose. Okay, cellulose. And with cellulose, it actually uses beta glucose, which is slightly different than the alpha glucose that we discussed or I talked about in glycogen and starch. Glycogen and starch is actually made with alpha glucose, but for cellulose, it's made with beta glucose, okay? And with beta glucose, you'll notice that each glucose molecule, like this one it's like upside down, the orientation upside down, and then this one is right side up, and this one's upside down. So notice how the glycosidic linkages and covalent bonds are like alternating. Right side up, upside down, right side up, upside down. So cellulose is very tough. It's a very tough polysaccharide, okay? In fact, most animals, we do not have an enzyme. We don't have the protein in our bodies to break down cellulose. There's no way for us to like break these bonds, okay? So oftentimes the cellulose in plants, okay, which make up the plant cell wall, the cellulose in plants is not digestible for us, and it's called fiber, okay? So you know how they talk about how we need to have a high fiber diet. That means it's a lot of plant material that we need to ingest. The cell wall, it's like the roughage, it's the vegetables, the fruits, okay? The cell wall is made up of cellulose, and it serves the purpose of providing structure for the plant. And for animals, when we eat plant material, it's very tough, and we cannot digest the cell wall. I mean, our teeth can mechanically open up the cell wall, like we can break open the cell walls with our teeth, but we can't really utilize it. We can't really digest it. It's just fiber that helps with digestion, fiber that helps with absorption of minerals and vitamins, and then it just basically exercises our intestines, and then we just poop it out, okay? We poop out any cellulose that is not digestible. So this is the difference between alpha glucose and beta glucose. So alpha glucose has this hydroxyl group that's facing downward from the first carbon, whereas beta glucose, this is the first carbon when you have the hydroxyl group on top, okay? That's the only difference between alpha and beta glucose. And notice in starch or glycogen, okay, the covalent bonds are in the same orientation, whereas starch, it's alternating orientation. And so when we look at the cell wall of plants, you have several layers of cellulose. It's not digestible. And then it's very rigid because you have several layers of cellulose, polysaccharide. And so it gives plants a lot of structure, okay? Like when you go outside and, I mean, you're being silly and you run towards a tree. I hope you never do that. But who's going to win? You or the tree, right? Obviously the tree. The tree is much more tough because there's so much cellulose, so much strength within the body of that tree, okay? If herbivores cannot digest cellulose, the cell walls, how do they get the nutrients from plants, okay? Well, this example of an herbivore is also called a ruminant, okay? A ruminant is a type of animal with like many rooms, okay? And you probably heard that cows have multiple stomachs. If you heard that, they're really referring to the rumen that's found in, or the rooms that are found in the digestive tract of cows. These rooms are where grass or the plant material is being digested by bacteria. Inside the gut, inside these rooms, inside the rooms of the cow are trillions, quadrillions, okay, of bacteria living inside of them. And the bacteria, they do have the enzyme. They have a way to break down cellulose. They have a mechanism for breaking down the cellulose. So the bacteria lives inside of the cow, and the cow is able to receive the nutrients from grass because the bacteria is there to help them, okay? And this special kind of relationship between bacteria and cows is called symbiosis, not symbiosis. It's called symbiosis, where they have a close relationship. And actually, because they mutually benefit from this relationship, it's called mutualism, okay, mutualism or symbiosis, where the bacteria is given a home, a place to live within the cow, and then the bacteria is breaking down the grass so that the cow can also get nutrients. So it's a very close relationship where they mutually benefit from this relationship. Also termites, you know how termites eat, wood, right? They're animals too, and they do not have a way to break down wood, but they have bacteria in their gut that's going to help break down the wood, the cellulose, because cellulose, again, is not digestible by animals. Okay, and then for structure in animals, so we talked about how cellulose is the structural polysaccharide in plants, but in animals, in some animals, not all animals, the structural polysaccharide is called chitin. Okay, chitin, and chitin looks like this, okay? You can see that it looks sort of like glucose, but there's an addition of a group of molecules, okay? And chitin is the structural skeleton. It's the exoskeleton. I should write that down. It's the exoskeleton of arthropods, and arthropods including insects, crabs, lobsters, okay? And chitin forms the exoskeleton of arthropods, and you can see here that this brown shell that's being left behind, this is where this cicada is molting and leaving behind its exoskeleton. It's made out of polysaccharide called chitin. And sometimes humans, we can, utilize chitin and make surgical thread or sutures. Okay, so that's carbohydrates. Now let's talk about lipids. Lipids are also macromolecules, or really large molecules, but we don't really consider them to be polymers because they're not really made up of long chains of anything. They're actually made up of like fatty acids, which are hydrocarbons, or just hydrocarbons in general, but they're not really made up of, like monosaccharides in carbohydrates. Okay, but the reason why lipids are grouped together as lipid is because they are non-polar, primarily because they're made up of hydrocarbons. Okay, so hydrocarbons are non-polar. They make up lipids. There's 3 types of lipids that we'll be talking about. There's more than 3 types of lipids, but in this discussion, we're just talking about 3. There's fat, phospholipids, and steroids. Those are the 3 that you need to remember. Okay, so fat. What is the function of fat? Think about a seal or a whale. They have a lot of fat, right? It's really for insulation. Okay, and then think about a bear that's about to go into hibernation. They just eat a ton of food and they get really fat. They will build up their fat storage. And what is that for? Energy storage. Okay, oops. Energy storage. So fat has a couple of purposes. One is for insulation, and then one is for energy storage. The fat molecule is made up of 4 parts. You have a glycerol. Okay, so 1 part glycerol, 1 glycerol, and then 3 fatty acids. And you can see here that these 3 fatty acid chains are made up of hydrocarbons, fatty acids. Actually, over here, part of the fatty acid, you see this is a carbonyl, and then this was actually a hydroxyl group that was part of glycerol. There's some oxygens there, okay? And we did mention how oxygens in a previous video or in class, oxygens are electronegative. You're going to end up with negative charges by the oxygen, and that's true. But the molecule is so big that you can, the negative charges on the oxygen is very minimal, and overall the molecule is nonpolar. Okay, so this is a nonpolar hydrophobic molecule called fat. And there's 2 types of fat. The first type of fat is saturated. And saturated fats usually come from animals. Okay, they usually come from animals. And the thing about saturated fat is that the hydrocarbons in the fatty acid chains are straight chains. Okay, they're straight. And because they're straight chains, they can be tightly packed together and form a solid. Okay, so one of the things about saturated fat is that it's solid at room temperature. Okay, at room temp. So fats from animals are going to be saturated fat, and they are solid at room temperature because the hydrocarbon chains in the fatty acid are straight. And the reason being is because in the fatty acids, every carbon has a single covalent bond. So it becomes a straight chain. And I'm not drawing every hydrogen, but you should know that carbon can form 4 covalent bonds. So those are where all of the hydrogens would go. And then this is attached to the glycerol over here. Okay, but it's the reason why you have straight chains is because there's single covalent bonds. So that's the first type of fat, saturated. The second type of fat is unsaturated. Okay, and unsaturated fats come from fruits and veggies, or I should just say plants, huh? Yeah, fruits, veggies, plant material. Okay, like olive oil comes from the olive fruit. Okay, you have like coconut oil. Actually, yeah, coconut oil, which is liquid at room temperature or when it's warm. Okay, so this is unsaturated fat, and the reason why it's liquid. So that's one thing that's important to remember. Saturated fat is solid at room temperature, whereas unsaturated fat is liquid at room temperature. It's because there is a double bond, a cis double bond between carbons. Okay, and in this example, see how it's sort of like bent? Okay, ooh. See how the fatty acid is bent? It's bent because there's a double bond between those 2 carbons. So if I just quickly draw what it might look like. So between these two carbons, okay, they form a double bond, and then it's going to cause it to bend, okay? And when it bends, they can't stack neatly on top of each other. And because they can't stack neatly on top of each other, they're going to be moving around each other as in liquid form, okay? So that's unsaturated fat versus saturated fat. Next, you have phospholipids. Phospholipids, the function of phospholipids is to make up the cell membrane. Okay, cell membrane. And it's made up of 2 fatty acid chains and then a phosphate group. Okay, so it's made up of 1 phosphate group. Okay, my writing is just horrible. And then 2 fatty acids. 2 fatty acid chains. Okay, I wish I could just type this. I don't know how to do that, but anyways. And it's going to make up the cell membrane. Phosphate group has lots of oxygens around it. That's going to be polar. Okay, that's going to be polar or hydrophilic. Remember, water loving. Whereas the tails are made out of fatty acid tails. They're non-polar or hydrophobic. So that's what's going on here with this phospholipid cell membrane. You have 2 layers, okay? You have 2 layers because the heads, which are hydrophilic or polar, they're facing inside the cell. So if this is inside the cell, there's water inside of your cells, and so they're facing inside the cell. And then this layer, the heads are facing outside the cell where there's water. Okay, and then the tails are facing each other. So this interior, cell membrane interior, is hydrophobic. Okay, all of this is hydrophobic, facing away from water. So the 2 layers is actually called a bilayer. Bilayer. Because bi means 2, layer referring to the 2 layers of phospholipids that make up your cell membrane. Okay, and so going back to this diagram, I kind of like this one because it shows you like if your cell was like a sphere, a circle, the heads are facing inward, the heads are facing outward. The phosphate heads are facing out and in, whereas the tails, the interior here, the interior of the membrane is facing, the tails are facing each other, okay? Okay, and then the third type of fat, or I should say lipids, are called steroids. And steroids have a very obvious structure where it's 4 fused carbon rings. 4 fused carbon rings. Like this is carbon ring number 1, 2, 3, 4. Okay, and actually this is a molecule of cholesterol. So this is a molecule of cholesterol, and you might be thinking of steroids. I thought steroids are like hormones. Well, actually, cholesterol that you might eat or make in your body is going to be used to make steroids or hormones like testosterone, estradiol, progesterone, okay? All of those hormones are actually made from cholesterol, okay? And you probably learned about the functional groups, right? The functional groups that are found here are going to give it a specific characteristic, okay? And all of these are hydrocarbons, right? That's why they are nonpolar or hydrophobic. All right, next topic is proteins. So there are several functions of proteins. You guys, proteins I would say is like the largest category of organic compounds, the largest category of macromolecules that make up living things, because proteins can do so many things. They're going to provide structural support. They're going to provide storage. Okay, they're going to provide transport for the cell or the body. They're going to be used for cell communication, for movement, defense against foreign substances. Okay, and when I say defense against foreign substances, like antibodies, you've probably heard of that before. Antibodies are proteins that attack foreign things like viruses and bacteria. Okay, but proteins do a lot. Like really quickly, structural support would be like collagen, okay? Collagen, elastin, that makes up your skin. A lot of these 2 proteins are found in your skin, giving you that youthful appearance, giving you structure, youth structure. Storage, like in eggs, there's albumin, which provide, it's storage within the egg protein that the little chick that's growing can use, okay? Transport would be like hemoglobin. Hemoglobin is a protein that transports oxygen throughout your whole body. Cell communication, we'll talk more about that in the future chapters, but like protein channels, okay, are going to be used to communicate between cells, okay? Movement, like your muscles. Oh, I should write myosin. Myosin, okay? Myosin is one of the, oops, I can't spell. Myosin, okay? It's one of the proteins that make up your muscles. And then again, defense against foreign substances would be antibodies. So those are just some examples of proteins in your body, okay? All right. Okay, next topic is, well, we're still talking about enzymes, sorry. We're still talking about enzymes or proteins. Okay, and in this slide, it just kind of went over what I went over, right? The different roles of proteins. Proteins do so many things. There's so many functions that they can have, and then these are just some examples. You know, one example of a protein that I didn't discuss on the previous slide are enzymes. Okay, proteins are, and there's a whole chapter devoted just to enzymes because they're so important. They help keep you alive. But when you eat food, you need to break it down, and there's no way to break it down except by hydrolysis using enzymes. Remember hydrolysis, breaking down molecules? Well, you need enzymes to actually help with the whole hydrolysis process, okay? And enzymes are proteins. They're going to help break down molecules. They're going to speed up the whole digestive process by breaking down the molecule. And so enzymes are proteins that are going to speed up reactions. And how do they do that? They're going to, like, if their job is to break bonds, they actually take in the molecule that needs to be broken and they physically put pressure on the covalent bonds and breaks it. Okay, it's like if I took this, marker thing and I, with all of my might, I tried to snap it in half, then it would break in half. That's kind of what enzymes do, is they take in the molecule and they put pressure on it and it breaks, okay? So here's our enzyme, and an example would be like sucrase, okay? Sucrase, I can't spell it. Sucrase is an enzyme that breaks down sucrose. I did mention sucrose earlier. If it ends in OSE, you should know that it's a sugar, right? Sucrose is the sugar that's found in your kitchen. Okay, it's the one that you bake with, you cook with, you add to your coffee, tea, okay? And it is a disaccharide, okay? It's a disaccharide. And in order to digest this disaccharide, you have to break this strong covalent bond. You need to break it into monosaccharides to digest it. Sucrase is the enzyme that does that, okay? See how this sucrase has this shape that fits this sucrose, okay? By the way, another word for the molecule that goes into the enzyme is called a substrate, okay? So the sucrose is a substrate. It's gonna fit right in here. It's like two puzzle pieces coming together. And when it fits in, the enzyme is going to put physical pressure on the molecule and it's going to break it. There's such a beautiful butterfly outside right now. Sorry, I got a little bit distracted. And so when it breaks it apart by hydrolysis, and so you can see that right here, water is coming in, water is going to break apart into an OH and an H, right? And they're going to bond with these monosaccharides. So you can see that here. These monosaccharides are going to form. Okay, so enzymes is one of the roles of, it's a type of protein that has this active site. I didn't mention active site. Okay, but where the substrate binds is called the active site. I should have mentioned that earlier. Okay, the active site is it's the place where they fit together, where the puzzle pieces fit together, where the substrate fits into to the sucrase enzyme. Okay, so proteins are polymers. Okay, and when we say polymers, that means that they're made up of monomers. All right, and the monomer of proteins are called amino acids. Amino acids. They make up long chains of proteins, okay, or what we call polypeptides, okay, polypeptides. Polypeptides, remember how like long chains of sugar is called the polysaccharide? Well, long chains of amino acids that make up proteins are called polypeptides. This is the basic structure of an amino acid, okay? Amino acids have a central carbon atom over here in the middle. They have a R group, okay, and I'll show you what R groups look like. You have the hydrogen atom attached to the central carbon atom. You have the amino group. Remember, that's a functional group that we went over that causes the molecule to be basic. And then here you have the carboxyl group, which causes the amino acid to be acidic, okay? So you have all amino acids have the same basic structure. They all have an amino group. They all have a carboxyl group. They all have the central carbon atom, which is attached to a hydrogen atom. And then they all have a specific R group. This is the basic structure of an amino acid that makes up protein. Okay? And when I said R group, there's actually 20 different kinds of R groups. There's 20 different kinds of amino acids based on the R group. So here's our amine group or amino group. Here's our carboxyl group. Here's our central carbon atom. Here's our hydrogen atom. Remember, those are the basic structures of amino acids. And then the R group, okay? The R group in this example, amino acid that's called glycine or gly for short, is a simple hydrogen atom. You guys, glycine is the most simple amino acid that makes a protein. The next most simple amino acid has an R group that is a methyl group. See this, CH3, that's a methyl group. That is going to make alanine, or ALA for short. Notice how all of the amino acids have the same basic structure. Amine group, carboxyl group, attached to a central carbon atom, attached to a hydrogen atom. Where they are different are right here, the R groups, okay? So when I say R groups, they are like different groups of atoms or molecules, I should say, okay? And all of these amino acids have different names. Val, Lu for leucine, isoleucine, Met for methionine, Phe, I guess that's how you say it, Phe, okay, for phenylalanine, and Trip for tryptophan. And these amino acids, how many are there? There's 9 of them. Okay, 9 of them are going to be nonpolar. And you might be wondering, Ms. Shin, why are they nonpolar? Well, when I look at them, most of them, not all of them, okay, are hydrocarbons. See that? Like this is a hydrocarbon. Hydrocarbon R group, hydrocarbon R group, hydrocarbon. So when we say nonpolar, we are not talking about the amine group or the carboxyl group. We're only talking about the R group. The R groups are nonpolar, okay? Because most of them here are hydrocarbons, which are, there's no charge, okay? Oh, there's a really pretty bird outside. Okay, and then you have the polar amino acids. It's not leaving, it's just like sitting right there outside my window. How cute. It's a black phoebe, if you were wondering. The name of the bird is Black Phoebe. Okay, and so when you look at this R group, this R group, this R group, this R group, what do they have in common? They're polar, okay, they're polar. Most of them have this oxygen. And then also, you know, nitrogen is also electronegative, okay? So, and sulfur we said is electronegative. So these, 6 amino acids, serine, threonine, cysteine, tyrosine, asparagine, glutamine, those are the 6 that are going to be polar, okay? And by the way, you don't need to remember the names of these amino acids. Just know that there's 20 of them, and some of them are nonpolar, like the ones on the 9 on the previous slide, and then these 6 that are polar, okay? So know that some are polar and some are nonpolar. And then there's 2 that are going to be acidic and 2 that are going to be basic. Sorry, 3 that are going to be basic. Electrically charged or ionic. Okay, we can call these ionic, we can call them acids, acidic, or basic. So this R group is acidic. And why is it acidic? Well, because this was a carboxyl. Remember carboxyl groups? C double bonded O, O, H. Okay, there used to be a hydrogen here. And that hydrogen popped off the molecule. It pops off the molecule. It's adding H plus to a solution. There was a H here, and now it pops off H plus into the solution. That by definition is what acids are. Acids are substances that increase the H plus in a solution. And it's all because of the carboxyl functional group, okay? Whereas these three are going to be basic, okay? These are the basic amino acids because their R groups contain an amine group, okay? And these amine groups are going to gain the H plus from a solution, okay? So see that? It went there, H plus went there, okay? H plus went there, and it's removing H pluses from a solution. By definition, that's what bases are. Bases are substances that remove H plus from a solution, decreasing the H plus concentration in a solution. So those are the 20 different amino acids that make up proteins. So then the next thing I want to draw is a peptide bond. You know, the monomers of proteins are amino acids. How do amino acids or the monomers even get together? Well, first of all, we know it's a dehydration synthesis reaction, right? So I'm going to go ahead and draw a, let's see, let me go ahead and draw an amino acid. I'm just going to draw R group like this. Okay, so that's an amino acid. That's my carboxyl group, amine group. I'm going to draw another amino acid here. Let me draw it like this. Okay, and then so if this is amino acid number 1 and this is amino acid number 2, how are they going to form a peptide bond? How are they going to form a covalent bond? How are they going to come together? Well, it's going to be by dehydration synthesis reaction, okay? This OH and this H come together, okay? They come together to make H2O. And then this carbon and this nitrogen, they form a covalent bond. Okay, so I've kind of run out of space, but maybe I'll just redraw it here using the same blue color. I'm just going to draw them attached by a peptide bond. Okay, CHRCOOH. Okay, so that's my peptide bond right here. This is my peptide bond, the bond between the carboxyl group, which is now missing the OH, and then the H in the amine group, okay? The N in the amine group. So this is a dipeptide. It is a, it's a polymer made up of just 2 amino acids, okay? And so the amino acid sequence that's written on this PowerPoint, it's just saying like you could end up with a long chain of amino acids. So when you have amino acids that are bonded by peptide bonds and you have a polypeptide chain, a long chain, a functional protein is never in a chain. Like you'll never find a functional protein that does its job in a straight chain, okay, a polypeptide chain. The protein is actually going to have 3-dimensional shape. Like in the picture at the top here, it says a ribbon model, and you can see that it's coiled like a ribbon, and that would be your polypeptide chain coiled. And then you can see the coil kind of folding, okay? So proteins are actually in 3-dimensional shape. They will never be functional if they are in a long, just straight chain, okay? I should have brought my example from school, and I left it at school, but it's basically a pipe cleaner with beads on it, and the pipe cleaner represents the chain of amino acids. Each amino acid would be a bead, okay? So I don't have that today. Instead, I just took a piece of string and I used marker to color in my amino acids. So I have a piece of string here with different colors, and each color represents an amino acid. So you will not find functional proteins in this state. You will not find them as a chain, a polypeptide straight chain. Instead, you'll find that the protein is going to form a coil, okay, form a coil. Like in this picture, the ribbon model, you can see it's coiled up, Okay, and the reason why it coils up is because there's hydrogen bonding that happens between R groups. Okay, and then this coil is going to fold even further. Okay, and that coil is going to fold even further until you get this three-dimensional structure of a protein that can actually do its job. Okay, so like in this picture here with the ribbon model, you could see it's coiling and then it's folding, okay, in different directions. And that's what a functional protein would look like. And when I say functional, remember how I said proteins do so many things, right? It can do its job if it's in this three-dimensional structure. And the reason why it's in a three-dimensional structure is due to the R group interactions. And we're going to be talking more about like the R group interactions when we go over the levels of protein structure, okay? So my straight chain, the amino acid sequence, okay, like my piece of string with all the amino acids linked together, that is the first level of protein structure or primary structure, okay? So you can see in this example that I have all of these amino acids, and there's 20 different amino acids, and they are abbreviated by the first three letters like lysine, valine, phenylalanine, glycine, arginine, cysteine, etc. You have, in this example, 129. I can see 129 amino acids linked together by peptide bonds, making a polypeptide chain. So the primary structure of protein is going to be held together by peptide bonds. Let me grab my pen. So peptide bonds, okay, that is what holds the polypeptide chain together. That's the primary structure of protein. Okay, I'm going to skip this one. We don't have too much time to talk about it. Next, like I said, proteins never stay in a chain. They're going to form what we call an alpha helix coil or a beta pleated sheet. Okay, so this is the alpha helix, or you can see the beta pleated, which means folding, sheet. Okay, sheets. So here is the amino acid. So I can see here that, let me see here. So this is the amine group. Okay, see that? This is an amino acid here. Amine group, central carbon atom, carboxyl group, and then this is the amine group of another one. Okay, so here is, these are chains of amino acids, okay? And they form a coil because they have hydrogen bonding between the amine groups and the carboxyl group. I can see that there's a hydrogen bond between this amine group and this carboxyl group. Do you see that right over here? The dotted lines represent the hydrogen bonds. So the hydrogen bonds hold the amino acid chain, this chain, into a coil shape, the hydrogen bonds. Same with the beta pleated sheet. They fold because there's hydrogen bonding between the sheets of amino acids. Okay, so that's what's going on in this diagram. You can see the coil and you can see the beta pleated sheets. The beta pleated sheet, not so obvious. So that was a secondary level. Primary was the amino acid sequence or the polypeptide chain, which doesn't stay like that, right? Proteins are going to either coil or fold. Folding and coiling is a secondary. And then tertiary, which means third, okay, tertiary structure of protein is where you have the coiling and the folding, folding even further. Okay, so let me just like kind of draw it for you here. So if this is my coil of amino acids and folding of amino acids, they're going to be held, or I should say folded even further. See how it like bends over here? How is it bending? Because of the R groups. The R groups are going to interact with each other. For example, right over here, this is one R group that's sticking out from an amino acid that's over here. I'm just going to write AA for an amino acid. Here's another amino acid over here. So let's say this is another amino acid. And its R group is over here. So these two R groups are attracted to each other because this is a hydrogen bond. This is a hydrogen bond. This is a positive hydrogen attracted to a negative oxygen. And opposite hydrogen, which is positive, is going to be attracted to the opposite charge, which is negative oxygen. They form a hydrogen bond, which causes this alpha helix and folding, which is the secondary level of protein structure, to fold on itself. So the tertiary is where the secondary folds on itself, okay? And then something else I want to point out to you, what's going on over here is that this is another R group of one other amino acid. Here's another amino acid, and this is the R group of that amino acid. Why are they facing each other? Because they're both hydrophobic. The R groups are hydrophobic. And proteins, proteins are in your cells, they're outside of your cells, and there's a lot of water in your cells, a lot of water outside of your cells. So if proteins are like floating around basically in a water solution, where are those hydrophobic R groups gonna go? Where are they gonna go? They wanna be tucked away from the water. So if there's water outside that surrounds this protein, there's like water all over the place that's surrounding the protein, those hydrophobic R groups are going to be tucked inside. They're going to fold inside of the protein away from the water. That's why this R group and this R group are tucked away, safe away from water, and we call that hydrophobic interactions, okay? And then why is this part of the amino acid chain folded inward? Well, there's another thing called a disulfide bridge or disulfide bond. It says bond right here. But here is one R group that contains a sulfur atom. Here's another R group that also contains a sulfur atom. So this is an amino acid. This is the amino acid. And by the way, the amino acid that contains sulfur is called cysteine. Okay, so that is actually, I think that's the only amino acid that contains sulfur. So this is a cysteine, this is a cysteine, this is the R group of one cysteine, and this is the R group of another, and they will form a covalent bond that's pretty stable. Okay, it's a stable disulfide covalent bond, disulfide bridge, linking this side of the amino acid chain to this side of the amino acid chain, causing it to fold even further. Okay, causing it to fold. If you have like really curly hair, the curls are held by disulfide bridges. Okay, let me just move me out of the way here. And then here's another example of why this polypeptide chain folds is because of an ionic bond. Okay, ionic bond. This is one R group. Here's another R group. And then let me just add, like, this is the amino acid. This is the amino acid. So those two R groups from those two different amino acids, they're attracted to each other. You have a positive charge, a negative charge, ionic bond, charge of attraction, right? So the tertiary level of protein structure is due to several types of bonds or interactions. You have hydrogen bonds, you have ionic bonds, you have hydrophobic interactions, you have disulfide bridges. Those are the four bonds and interactions that cause the coiling or the folding, which is the tertiary structure, to fold even further, okay? So you got the primary level of protein structure, which is just the straight chain. Then you have secondary, which is the coiling or folding, and then you have tertiary, which is coiling and folding even further. And then some proteins, not all, but some proteins that are actually functional. In order to be functional, you have to have two or more tertiary structures coming together. So quaternary, which means fourth level, is two or more tertiary structures coming together. Okay, so here is collagen, which I mentioned is a structural protein in your skin. It's actually made up of 1, 2, 3 polypeptide chains. Okay, and then hemoglobin, a really important protein that delivers oxygen to all of your cells. There's actually 1, 2, 3, 4. There's 4 tertiaries together. Okay, so that's quaternary structure protein. Again, not all proteins in order to be functional are quaternary, but these are just two examples of functional proteins that actually do a job, but in order to do the job, you need to have 2 or more. Collagen needs 3, hemoglobin, you need those 4 subunits. So this is a nice summary of the 4 levels of protein structure. Primary, which is just the chain of amino acids. Secondary, which is the helix, which is the coil or beta-pleated sheet. And then tertiary is where you have the coiling and beta-pleated sheet, which is secondary, folded further on itself. And remember, there's 4 types of bonds that are responsible for this. You've got the ionic bonds, the hydrogen bonds, hydrophobic interactions or disulfide bridges. And then quaternary structure is where you have 2 or more of these guys, tertiary, coming together. Oh, and I forgot to mention that the quaternary structure of protein, how are they held together? Same way as the tertiary. disulfide bridges. They're held, these four in this picture, I think it's four. These are held together by disulfide bridges, ionic bonds, hydrogen bonds, and hydrophobic interactions. Okay, should I go over this? So an example, yeah, I'll just go over an example of like a mutation where there's a change in the primary structure, like let's say that the amino acid changes, would be sickle cell anemia. So sickle cell anemia is a genetic disorder in the hemoglobin protein. The hemoglobin protein, instead of having a, right over here, a hydrophilic, okay, instead of having a hydrophilic, which is glutamine, hydrophilic amino acid, it's replaced by mutation. It's replaced by a hydrophobic amino acid. So we're going from hydrophilic to hydrophobic. Do you think that's going to change the way that the protein folds? It sure is, because remember, these proteins are surrounded by water. So if you're going from hydrophilic, which can interact with the water surrounding the protein, to hydrophobic, which means that it's going to fold differently. It's going to tuck, this valine is going to be tucked inside rather than outside. And so it's going to change the shape of the protein. And then if you change the shape of the protein, the hemoglobin protein, you're going to change the shape of the cell. Okay, because the hemoglobin is found inside of red blood cells. And if it's misshapen, you're going to end up with sickle cell anemia. Okay, so these are red blood cells that are shaped irregularly versus regular round shape, like donut shape, red blood cells. And this is all due to a change in the primary structure of protein. Okay, so denaturation. What is denaturation? This is when there's factors in the environment that can change the shape of the protein. Let's just ignore this meme in the background for now, okay? Some things that can affect the protein structure is changes in pH, changes in salt concentration, changes in temperature, and we'll be talking more about this when we go over enzymes, okay? But if this is the normal protein, right? And by the way, what level of protein structure is this in? Is this primary, secondary, tertiary, or quaternary? I hope you said tertiary, because I can see coils, right, folded upon itself. I don't see more than one, I just see one. So this is tertiary structure of protein. If you heat up this protein, if you change the temperature, heat it up, it's going to break those hydrogen bonds. It's going to break those ionic bonds, okay? It's going to lose its shape. Denaturation means to lose shape, okay? And so the protein is now changed shape. It's no longer in the appropriate functional three-dimensional structure. It's now in this loose structure. And in order to do its job, the protein has to stay at its normal shape to do its normal function. And so proteins can easily be denatured by changing temperature, by changing the pH. Like if you make something too acidic or too basic, then the proteins that are naturally in like maybe a pH of 7, a neutral pH, if you put it in acid, then it's going to denature, okay? So a good example would be like cooking, like when you cook eggs, right? The white part of the egg is mostly protein. It changes shape and it turns opaque. It goes from clear to opaque. The last type of macromolecule would be nucleic acids. Then there's two types of nucleic acids. There's DNA, which is deoxy, and I'll try to write neat. Deoxyribonucleic acid, right? DNA. And then RNA, ribonucleic acid. Okay, RNA. So what is the function? For DNA, what does the DNA do? It basically holds genetic information. Okay, it holds genetic information. What does RNA do? RNA has so many jobs, okay? RNA is like the busybody nucleic acid, whereas DNA just kind of sits in the nucleus, okay? RNA is responsible, it plays a big part in protein synthesis, okay? It plays a really big part in protein synthesis. Like we'll talk about the different kinds of RNA, like mRNA. So maybe I'll just write a few examples. mRNA, tRNA, rRNA, they all play a big role in protein synthesis, okay? And what makes up nucleic acids are going to be nucleotides, okay? And this is in regards to protein synthesis, which RNA does, okay, and DNA holds the genetic material. It's called the central dogma of biology. That term actually was invented by James Watson, who helped discover the structure of DNA. And he was basically saying, like, all of biology boils down to this, okay? That from DNA, you get a copy of the DNA in RNA, and then that RNA can be used to make protein, and you're made up of protein. Like, organisms are made up of protein. And so where does it all come from? It comes from this process of taking genetic information, copying it into RNA, and then using RNA to make protein, to make up organisms, okay? More on this, we're going to be talking about the central dogma in greater detail later on. But again, what are nucleic acids made of? What is DNA and RNA made up of? Nucleotides. They are the monomers of nucleic acids. Just like how with carbohydrates, it was the monosaccharides. With proteins, it was the amino acids. Okay? So you guys remember that. Nucleotides are the monomers of nucleic acids. And this is what a nucleotide looks like. Okay? The nucleotide has three main parts. You have the phosphate group, you have a pentose sugar, pent meaning 5 carbon sugar, and then you have a nitrogenous base, a carbon ring that contains nitrogen. Okay, and you can see that the nitrogenous bases, these are the nitrogenous bases. There's 5 different kinds of nitrogenous bases. Nitrogen containing base. You can see it's a carbon ring with nitrogens thrown in. Okay, carbon ring with nitrogens. So those are what we mean by the nitrogenous base. You know, there's 2 kinds of nitrogenous bases. You have the pyrimidines, and then you have the purines. Pyrimidines are the cytosines, thymine, uracil. Purines are adenine and guanine, okay? In DNA, that sugar, that pentose sugar in DNA, it's called deoxyribose. In RNA, it's called ribose, okay? The only difference between deoxy and ribose is right over here. See this? Attached to carbon number 2, this is carbon number 2, there's a hydrogen atom in deoxyribose. In ribose, there's a hydroxyl. So deoxy means missing oxygen, okay? And it's referring to carbon number 12 is missing an oxygen, okay? And then the phosphate group. Phosphate group is, as you guys can see here, it makes the DNA molecule negatively charged. There's a negative charge on the phosphate group, so DNA is negatively charged. So when we compare DNA and RNA, the nitrogenous bases in DNA are going to be adenine, thymine, guanine, and cytosine. Okay, so let me just quickly go back here. Okay, it's going to be adenine, guanine, thymine, and cytosine. Okay, whereas in RNA, it does have the adenine, but it doesn't have a thymine. It has a uracil, guanine, and cytosine. So going back, there is in RNA uracil. There's no thymine in uracil. Instead, it's going to be, there's no thymine in RNA. It's uracil. Okay, and then the sugar in DNA is deoxyribose, right? Deoxyribose. Whereas the sugar in RNA is ribose. Structure. What does DNA look like? It's actually a double helix. Okay, it's double-stranded, and then helix meaning that it's coiled. Okay, RNA is single. It's single, like some of you. It's single and it's single-stranded. Okay, it's not in a helix. The role of DNA is to hold genetic information. Or I should just write genes. Okay, info. Whereas RNA it plays a big role in protein synthesis. Okay? Protein synthesis. All right. I think, yeah, so DNA, you got the double helix, 2 strands, one on the left, one on the right here, and then it twists, okay, into a helical form. And that's where the genes are found. Genes are found on the DNA. And there's complementary base pairing. What do we mean by complementary base pairing? On one strand, so this is one strand right here, and here's the other strand of DNA. Adenine, that nitrogenous base adenine, it pairs up with thymine on the other strand, and they form a hydrogen bond. They form two hydrogen bonds. Between guanine and cytosine, there's three hydrogen bonds. So you can see here that for every T and for every A, they come together. For every G and every C, they come together. So that's what we mean by complementary base pairing. The two strands of DNA, they actually can come together like magnets, hydrogen bonding. They're attracted to each other. They like magnets, they come together. The two strands can come together. A and T always come together, and G's and C's always come together. Okay, and DNA is anti-parallel, okay? And we're not going to go into too much detail about this, but the anti-parallelness of DNA refers to, okay, the carbon ring in the ribose, in the 5-carbon sugar, sorry. The ribose or the DNA. Actually, it's deoxyribose, right? So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and then over here in this point, it's carbon number 5. Carbon number 1, 2, 3, 4, 5. And then down here, I'll just go 1, 2, 3, 4, 5. And so when we say the anti-parallel nature of DNA, let me just go back here. This is carbon number 1, 2, 3, 4, 5. And then down here, 1, 2, 3, 4, 5. So anti-parallel means that the two strands of DNA are parallel. That's what the parallel means. They're side by side, but anti means they're going in opposite directions. They're going opposite directions. What do we mean by that is the sugar. The 5-carbon sugar is closest to this end. So we call this end of DNA 5' because it's referring to the closest carbon. The carbon number 5 is closest to this end. Which carbon is closest to this end? The 3rd carbon, which is 3'. So this strand of DNA is 5 on one end and 3' on the other. Whereas look at this strand of DNA. This is going the other direction. Okay, this is carbon number 1, this is carbon 2, this is carbon 3, this is carbon 4, this is carbon number 5. The closest carbon to this strand is 3. See that? Carbon number 3, this is the 3' end. Look at the other sugar on the opposite side. Carbon 1, 2, 3, 4, 5. Carbon 5 is closest to this end, down at the bottom. So DNA is, you have the two strands that are parallel, but they're going in opposite directions. And when we say direction, we're just referring to the 5' and 3'. You have one DNA strand, 5' here, 3' there. The other strand is 3' here, 5' there. So they're like opposites, okay? Opposite direction. Okay, and then sometimes DNA can be used as a tape measure for evolution. Like if you see similarities between different species of animals, you can say that they're closely related, okay? So when you look at the DNA sequence, right? So going back to here, this picture, like this is the sequence, A, G, T, A, C, G. That's a DNA nucleotide sequence. If you look at the DNA sequence of humans and other animals and find similarities, then you can say that they're like related, okay? So it can be used as like an evolutionary tape measure. And I think that's the end of our presentation. All right, I hope this wasn't too long. This is a really big part of the AP exam. You really do need to know about the 4 different types of macromolecules or organic compounds that make up living things. You first have the carbohydrates, you have the lipids, you have the proteins, and then you have the nucleic acids. So those 4, okay? All right, I hope that this video was helpful for you. I know I apologize, I'm kind of tired, but you know, I hope that it was helpful and I wasn't too boring. All right, have a good day. Bye.